It's June 16th, and this is your daily brief in science. Here's everything you need to know. Researchers are delving into the cancer defenses of elephants and similar species, aiming to uncover new treatments that could bolster human resilience against cancer. This investigation challenges Pato's paradox, which posits that larger animals should have higher cancer rates due to their greater number of cells and longer lifespans. A pivotal 2015 study revealed that elephants possess 19 extra copies of the TP53 gene, a critical component in detecting DNA damage and triggering the death of potentially cancerous cells. The recent research analyzed nearly 300 animal species and over 16,000 necropsy records, discovering that various species, including certain birds and bats, have evolved unique cancer resistance mechanisms. Interestingly, the findings suggest that elephants are not alone in their cancer resilience, as other species also demonstrate low cancer rates. In addition to elephants, certain birds, bats, and lizards exhibit low levels of cancer, while ferrets and opossums show higher rates. The study further revealed that animals in captivity do not experience higher cancer rates solely due to longer lifespans compared to their wild counterparts. Contrary to expectations, a larger body mass was linked to a slightly elevated cancer risk, while longer gestation periods were associated with lower cancer risk, likely due to enhanced cellular safeguards developed during fetal growth. Overall, while the study indicates a slight increase in cancer risk with larger body mass, the correlation is not as strong as previously believed. This emerging field of comparative oncology aims to uncover cancer-resistance strategies across species, with the ultimate goal of translating these findings into medical advancements for humans. Notably, while the TP53 gene is crucial for cancer prevention in humans, we possess only 2 copies, in stark contrast to elephants' significant surplus. Researchers at Stanford University have unveiled a groundbreaking device known as the Milli-Spinner, designed to effectively remove blood clots that lead to ischemic strokes. This innovative mechanism not only extracts clots, but also reshapes and compacts them, with the potential to transform thrombectomy procedures and enhance patient outcomes. The Milli-Spinner operates through a catheter, utilizing a hollow tube that rapidly rotates while employing localized suction to shrink clots without causing rupture. Traditional thrombectomy methods often lack precision and have a low success rate, typically managing to remove clots on the first attempt around 50% of the time. In contrast, the Milli-Spinner boasts an impressive first-attempt success rate of 90% for the most challenging clots, significantly outperforming existing devices, which have only an 11% success rate. According to neuroimaging expert Jeremy Haidt, the Milli-Spinner has more than doubled the efficacy of current technologies, achieving a remarkable 90% success rate on difficult clots. The device is capable of reducing clots to as little as 5% of their original size, allowing red blood cells to flow freely while compact fibrin balls are extracted. The research team is also exploring other biomedical applications for the Milli-Spinner, including the potential for kidney stone removal. To advance this technology, the researchers have established a company to license the Milli-Spinner and plan to initiate clinical trials soon, seeking approval for patient use. Senior author Renee Zhao emphasized that current thrombectomy technologies cannot effectively reduce clot size, whereas the Milli-Spinner applies compression and shear forces to achieve this. The concept for the Milli-Spinner is rooted in Zhao's previous work on milli-robots, which were initially intended for medical applications, and evolved into this advanced clot removal technology. With a 15% failure rate in current treatments, the Milli-Spinner aims to significantly improve these statistics, offering hope for better outcomes in stroke treatment. A recent study from the University of O'Higgins reveals that mastodons, which vanished from South America around 10,000 years ago, played a crucial role in the seed dispersal of large fruited plants. This research provides the first direct fossil evidence of frugivorous behavior in Nochiomastodon platensis, based on a detailed analysis of 96 fossil teeth collected from southern Chile. Published in Nature Ecology and Evolution, the study highlights significant findings from Lake Tagua Tagua, where the fossil teeth were analyzed. Isotopic analysis and fossil calculus studies confirmed that mastodons consumed fleshy fruits, such as those from the Chilean palm, indicating their vital role in forest regeneration. Furthermore, stable isotope analysis indicated that mastodons inhabited a fruit-rich forest ecosystem and traveled long distances, facilitating seed dispersal. The extinction of mastodons disrupted a coevolutionary alliance, leading to an increase in the threat level of megafauna-dependent plant species in central Chile, where 40% are now at risk. Species such as Gomortega, the Chilean palm, and the monkey puzzle tree are now found in fragmented populations with low genetic diversity, highlighting the long-term effects of mastodon extinction. The findings underscore the importance of paleontology in understanding contemporary ecological crises, emphasizing the need to recognize and preserve what remains. The study also validates the neotropical anachronisms hypothesis proposed by Daniel Jansen and Paul Martin in 1982, suggesting that large fruits evolved to attract large animals like mastodons for seed dispersal. Current conservation efforts are hindered by the loss of ecological relationships established over millennia, with species like Gomortega and the Chilean palm surviving in fragmented populations. Led by Dr. Erwin Gonzalez Guarda, the study involved specialists in paleodiet and paleoenvironmental reconstruction from institutions including IPHES, CERCA, and the Universitat Rovira i Virgili, illustrating that understanding past ecological relationships is crucial for addressing contemporary ecological challenges. Top U.S. health officials are raising alarms about the future of mRNA vaccines, citing concerns over pandemic preparedness and the potential impact on other vaccine types. This comes in the wake of significant changes within the Department of Health and Human Services. On June 12, 2025, Robert F. Kennedy Jr., the newly appointed Secretary of Health and Human Services, disbanded the Independent Advisory Committee on Vaccines for the CDC, a move that has sparked worry among public health experts. Kennedy's controversial research initiatives may lead to legal challenges against vaccines, which could disrupt the Vaccine Injury Compensation Program. This raises questions about the future of routine immunizations and the willingness of manufacturers to remain engaged in vaccine development. Additionally, the Trump administration's cancellation of a $766 million contract with Moderna for developing mRNA vaccines against pandemic flu viruses has raised significant concerns about U.S. biosecurity. Historically, the U.S. has focused on strengthening its pandemic defenses since the George W. Bush administration, particularly in rapid vaccine production and public health readiness. However, experts warn that if a bird flu pandemic were to occur soon, the U.S. would be inadequately prepared due to recent cuts in health funding. HHS Communications Director Andrew Nixon stated that the decision to halt mRNA vaccine funding was based on ethical and scientific concerns, though critics argue this reasoning is unfounded. Flu pandemics have historically resulted in millions of deaths globally, highlighting the critical importance of preparedness against emerging viral threats. Experts emphasize the speed and adaptability of mRNA technology, which contrasts sharply with traditional vaccine methods that are slower to develop. Growing concerns indicate that the U.S. may have to rely on foreign countries for vaccines in a future pandemic, which could lead to delays similar to those experienced during COVID-19. Furthermore, anti-vaccine sentiments have surged during the pandemic, with figures like Kennedy promoting misinformation that questions vaccine safety. This complicates public health efforts and poses a challenge to maintaining trust in vaccination programs. mRNA vaccines have represented a significant breakthrough in vaccine technology, playing a crucial role in combating the COVID-19 pandemic and showing promise for treating various diseases. Groundbreaking research on axolotls, a type of aquatic salamander, has unveiled crucial insights into their remarkable regenerative abilities. Conducted by James Monahan, a professor at Northeastern University, the study published on June 10, 2025 in Nature Communications, highlights the role of retinoic acid in guiding injured axolotl cells on which body parts to regenerate. Monahan's research specifically identifies retinoic acid as a key signaling molecule in the regeneration process. This compound allows cells to determine their location within the limb, influencing the extent of regrowth. For instance, higher concentrations of retinoic acid in the shoulder prompt the complete regrowth of limbs, while lower concentrations in the hand result in only a hand regrowing. The study also pinpointed an enzyme called CYP26B1, which regulates the breakdown of retinoic acid, playing a vital role in controlling the regeneration process. When CYP26B1 was blocked, it led to excessive limb growth, underscoring its importance in limiting regeneration. Monahan's manipulation of retinoic acid levels in axolotls resulted in the growth of duplicate limbs, demonstrating the potential to control regenerative processes. To study the activation of retinoic acid signaling in injured cells, the team utilized genetically modified axolotls that glow fluorescent green. This research opens up possibilities for human medicine, suggesting that similar mechanisms could allow for healing without scarring or even regrowing fingers and limbs. While human limb regeneration remains a distant goal, understanding retinoic acid signaling in axolotls may lead to advances in healing and scar prevention. Catherine McCusker, an associate professor not involved in the study, emphasized the importance of ongoing basic research in biology to uncover novel regenerative techniques. Monahan envisions future gene therapies that could enable human cells to respond to regeneration cues by activating or deactivating specific genes at the right times, potentially leveraging CRISPR technology. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.